So in this presentation, we're going to actually look at the chemistry of the reactions of the citric acid cycle, focusing on the functional group transformations and the reaction types. Right? We won't be doing the enzymology. Right? We're not going to look specifically at the enzyme chemistry of these reactions. We'll save that for another presentation. In a previous presentation, we talked about the decarboxylation of pyruvate to make acetyl-CoA. So now we're going to focus on the actual entry into the citric acid cycle. So we'll start with the citrate synthase reaction, where we combine oxaloacetate with acetyl-CoA to make citrate. So here we are focused in a little bit more. Right? So here is our acetyl-CoA, right? So here's our two new carbons coming in, right? Here's our backbone of our oxaloacetate. And so when we bring this together, and the reaction occurs in a regiospecific way, so that the two carbons of the acetyl group from acetyl-CoA are there, and the four carbons of oxaloacetate are there, where this carbon here is this carbon right there. So here we are, focusing in a little closer, right? So we said that, right, these two carbons become these two carbons of the product, and these four carbons become these four carbons of the product, right? And that it's this carbonyl carbon that becomes that carbon there. So we're going from a ketone to a tertiary alcohol, which means there was an addition, right? So we've got a thioester aldol. Instead of a condensation, I think it would be more proper to replace this with addition, right? It is an aldol addition. And we note, right, one of the things that we can note is that this reaction is irreversible. Right? This was one of the irreversible reactions that gives the citric acid cycle direction. Well, we know that hydrolysis of a thioester releases a lot of energy. So in this case, it is the hydrolysis of this thioester bond to release the free thiol as coenzyme A. It's that energy release that ultimately makes this irreversible. So we've got a thioester aldol addition, right? So we're adding this carbon across the carbonyl to make a tertiary alcohol. And to make the whole thing energetically favorable, we're hydrolyzing that high energy thioester bond. So now we will move to the Econotase reaction. And the Econotase reaction occurs in two steps. because we want to convert this tertiary alcohol. And remember that a tertiary alcohol can't be oxidized. And we're going to convert that into a secondary alcohol, which I can write today, can be oxidized. So here we're focusing in on that reaction, and we can see that the chemistry is going to go on right here, right adjacent to the tertiary alcohol. But remember, it is this part of the molecule that came from the acetyl-CoA. So this is not affecting the new group that we added from acetyl-CoA. And so we said this reaction occurs in 2 steps. So there is an elimination reaction that removes water and turns that into an alkene that is conjugated with both the carbonyl of this carboxyl group and the carbonyl of this carboxyl group. So that is a very stable alkene, right? Because it is stabilized by resonance throughout that structure. And that structure is something called econotate. Now, econotate here is an enzyme-bound intermediate. That's what the bracket means. It's not released from the enzyme. And then we bring water back in, and we add water back across the double bond. But in this case, we do it in an anti-Markovnikov orientation, so that we end up then with a secondary alcohol instead of that tertiary alcohol group that we had in the beginning. In our next reaction now, since we've made this secondary alcohol that can be oxidized, and we know that one of the things that NAD is good at oxidizing is alcohols, we can use isocitrate dehydrogenase. But interestingly, we get an oxidation and a decarboxylation. Right? We're going to lose carbon dioxide. That carbon dioxide is going to come from that group right there. To go from the 6-carbon molecule isocitrate to the 5-carbon molecule alpha-ketoglutarate. In this reaction, note, is shown in only one direction. It is one of our irreversible steps. And what makes it irreversible is the loss of this carbon dioxide as a gas, right? It makes the whole reaction metathetical, right? Something is leaving the reaction by taking itself out of the reaction mixture. In this case, the carbon dioxide leaving as a gas. So here's our reaction, right? So here is that secondary alcohol group that NAD can oxidize, right? So NAD does its oxidation, takes electrons in the form of that hydride ion, right? That atom comes off as a hydrogen with its 2 electrons. And so we end up then making this new ketone group. But that gives us a structure that we call an alpha carboxyl ketone, right? Where, right, this is the carboxyl group. And alpha carboxyl ketones are susceptible to spontaneous decarboxylation. Right? And so if we can decarboxylate and then protonate the product, then we get alpha-ketoglutarate, right, where we've lost that CO2 at that position. When we get to alpha-ketoglutarate, we've created a molecule that is a carboxyketone. If we look at pyruvate, it is also, right, a carboxyketone, right? We've got, right, this structure here, which is mirrored in this structure here. And so the structure of alpha-ketoglutarate has that same functional group arrangement that pyruvate has. And so we can oxidize it in the same way. So there is an alpha-ketoglutarate dehydrogenase, which has the same three activities that we had in pyruvate dehydrogenase. There is a decarboxylation, right, where carbon dioxide is removed, and that is that group there. The remaining acyl group, if we go here, right, the acyl group is transferred to coenzyme A, but that results in reduction of the essential prosthetic group coenzyme, and so then we have to oxidize that to return it to activity. Our result then is the same kind of high energy thioester linkage that we got in acetyl-CoA, in our product succinyl-CoA. Okay, so the chemistry of alpha-ketoglutarate dehydrogenase is the same as the chemistry for pyruvate dehydrogenase. And as we saw in the previous reaction, it is the loss of this carbon dioxide as a gas that makes this whole process irreversible. And so we see here it has the same kind of organization. It has the alpha-ketoglutarate dehydrogenase component, E1, with thiamine pyrophosphate, right? That's responsible for the decarboxylation reaction to isolate the acyl group. Then the acyl group is transferred first to the lipoamide of E2, and then to coenzyme A, and then the dihydrolipoyl dehydrogenase uses FAD to oxidize the lipoamide to return it to the proper oxidized form for activity there, and then NAD is used to oxidize the FAD. As a matter of fact, right, this subunit and this subunit, these are shared with pyruvate dehydrogenase. right? It's the same protein used in both places as part of the multi-enzyme complex. This alpha-ketoglutarate dehydrogenase subunit is unique to the alpha-ketoglutarate dehydrogenase complex because we have to recognize a different substrate, alpha-ketoglutarate instead of pyruvate. And note the other difference here is that there are no controlling kinase or phosphatase. And since, right, in pyruvate dehydrogenase, those interacted with E1. And so that's another difference. So the E1 of alpha-ketoglutarate dehydrogenase recognizes a different substrate, and it is not controlled by a kinase phosphatase system, so there's no recognition sites for those interactions. But otherwise, the structure of the alpha-ketoglutarate dehydrogenase complex is like the structure of the pyruvate dehydrogenase complex. So here is that high-energy thioester in succinyl-CoA, and we're going to accomplish here, this is the one time we directly make a high-energy phosphate molecule, right? Again, in what we call a substrate level phosphorylation reaction. Right? So before we used our thioester to make the citrate synthase reaction irreversible. Here we're going to use the energy of our thioester to actually make a high-energy phosphate molecule. And so here's a layout of the reaction of the succinyl-CoA synthetase. It's also sometimes called succinate thiokinase. And so here we have our high-energy thioester, and we're going to use a phosphate ion as the nucleophile to attack that in a nucleophilic acyl substitution reaction with the coenzyme A acting as the leaving group. And so we get an enzyme-bound succinyl phosphate that is now a mixed carboxylic phosphoric acid anhydride, which we know is also a high-energy molecule. So then a histidine from the enzyme acts as the nucleophile to attack the phosphate so that the his takes the phosphate and then the succinate can act as the leaving group. Right? So succinate acted as the leaving group. And so we're left with a phosphorylated histidine. GDP can come in as the nucleophile, take the phosphate, making GTP. There's our substrate level phosphorylation. That returns our enzyme to the free histidine. That can come back here, right, to continue the process. And then our nucleoside diphosphate kinase can transfer the high-energy phosphate from GTP to ATP. And so we get a net synthesis of ATP right? And so this is true in animal systems. We go GTP and then nucleoside diphosphate kinase to ADP, right? In plants, the succinyl-CoA synthetase just uses ADP, right? It skips the GDP step. So as a result here, right, we have used the energy of our high energy thioester to generate ATP in the mitochondrial matrix. Right? So now we're here at succinate, and so we're going to take these two saturated carbons and make a transalkene. And that is an oxidation that can be done using FAD. We won't worry about the specific mechanism of the reaction, right? But we've got these two sp3 hybridized carbons. When we oxidize them, that is when FAD is used to oxidize it so that we make FADH2, right? We result in the formation of an alkene that is conjugated with both carbonyl groups, and that is fumarate. One other thing that is unique about succinate dehydrogenase is that succinate dehydrogenase is an integral protein of the mitochondrial inner membrane. The other enzymes of the citric acid cycle are soluble proteins of the mitochondrial matrix. And so this is going to be important because this then lets it interact directly with the electron transport mechanism. Right, which is different from the other enzymes of the citric acid cycle. So we're here at fumarate, and so the next thing we're going to do is convert our alkene into an alcohol by doing an addition reaction of water. And that's going to be catalyzed by the enzyme fumarase. So here is our fumarase reaction. Right? We're going to add water to go from an alkene to a secondary alcohol. Right? We've added water across that double bond. We're not going to worry about the enzymology for the moment, but if we wanted to characterize this addition, right, it is a conjugate addition. Right? So if you go back to your organic chemistry, right, it is a conjugate addition influenced by the presence of the carbonyls next to the double bond. Now that we've made a nice secondary alcohol, we can oxidize it. And so when we oxidize malate to oxaloacetate, oxaloacetate was the molecule that condensed with acetyl-CoA in the first place. And so this oxidation will complete our cycle. So here's our malate, right? The important functional group here is this secondary alcohol. We know primary and secondary alcohols can be oxidized by NAD in a very straightforward way, right? To make NADH. and then that converts the alcohol into the carbonyl, and that is oxaloacetate, where we started our cycle. So we've gone all the way around the cycle now. Oxaloacetate can react with another molecule of acetyl-CoA to start the cycle again. So let's look at the carbon flow in the cycle, right? So if we highlight our carbon dioxide carbon and our acetyl group, right? In the pyruvate dehydrogenase reaction, it is that carboxyl group that is lost as carbon dioxide, and the two carbons of the acetyl group are transferred to acetyl-CoA. In our citrate synthase, right, we noted earlier, right, if this is the 4-carbon backbone of our oxaloacetate, those 4 carbons end up there, and the 2 carbons from acetyl-CoA end up there. And remember that the enzymes, even though this is an achiral molecule, the enzymes can recognize the difference between those two ends. And we used Econotase as an example in our enzyme chemistry chapter of that recognition, being able to recognize separately the two ends of a non-chiral molecule, because the Econotase is chiral because it's made of chiral amino acids. Right? And so we remove water, we put water back, but still the enzymes can tell the difference. Right? Those 4 carbons still came from oxaloacetate. Those 2 carbons came from acetyl-CoA. Now, the carbon dioxide that we lose, right here, is this carboxylate group right there. So that first carbon dioxide that we lose started as this carbon in oxaloacetate, right? So the first carbon dioxide that comes off is carbon that was in oxaloacetate, not carbon that was in acetyl-CoA. And then when we do our alpha-ketoglutarate dehydrogenase, again, right, here we go. Right, we've got now these 3 carbons left that came from oxaloacetate. These 2 came from acetate, and it's going to be this carbon. Right, let me do it this way. This carbon that's going to come off as that carbon dioxide. So that again is coming off, and it started as a carbon in oxaloacetate. Right? That one, right? And it was actually that carbon of oxaloacetate. Right? If we follow that carbon through. And so when we get to the level of succinyl-CoA, right, the 2 carbons closer to the thioester came from oxaloacetate, and the other 2 carbons came from acetyl-CoA. And so when we make succinate, that same thing is true. But the problem is succinate is a symmetrical molecule. And so at this point, the molecule is able to flip over if it wants to, right? There's no way for the enzymes to tell one end from the other. And so we lose the ability to tell one end from the other. And we end up then with all of those carbons appearing as if they came from the same source. Right? And so we can't tell which end is which as we go from succinate to fumarate to malate. Right? It isn't until we get back to oxaloacetate that we're asymmetrical and dealing with a reaction that can, that is directing to one end of the molecule or the other. So we need to think about the carbon flow through the process as well. So that is an overall look at the chemistry of the citric acid cycle. You should be able to recognize the functional group transformations and understand the relationship so that if I gave you these structures out of order, you can understand how one functional group is converted to another to put them back into the proper order.